So next up, we have the enigmatic Dave Beier talking about one of our favourite topics, a little bit of epistasis.
Okay. As many of you know, the focus of the research in my lab is using forward genetics, specifically ENU mutagenesis, in order to find defects of organ development, ideally as models of human birth defects. And this has worked very well. We're screening for sort of gross morphological abnormalities, and we see these, and these days it's straightforward to do positional cloning and characterize them. So this approach has been successful, but what we're trying to do is push this forward mutagenesis strategy to ask more subtle questions, to be more sensitive. And one of the ways that we've been doing that is to include reporter genes that identify specific anatomical structures and look for abnormalities in those, which enables us to detect defects in the context of what is a grossly normal mutant mouse. This is our first study done by Noelle Dwyer when she was a postdoc with Chris Walsh, using a very robust reporter that identified axonal tract projections. But what was actually more interesting to us was to look at an interesting aspect of the mouse cortex, which it shares with the human cortex, which is its 6 layers, a laminated structure. And this is a slide from a study from the Allen Brain Institute, in which they've actually marked the layers using different in situ hybridization of different genes, and it demonstrates both the anatomical layering, but also the presumptive functional layering that exists, since these layers are expressing different markers. And what was important to know is that this is developmentally regulated, and there is a well-characterized mutation in the mouse, the Relr mutation, originally identified, as you imagine, because it's ataxic, and it lacks a cerebellum, but what also it has is it has a profound disorganization of this layered structure. So what we were interested in doing is finding other genes that might affect this layering. And for this purpose, Rolf Stottman, when he was in the lab, identified some candidate reporter loci and imported this one, RGS4. This worked great for our purpose, or seemed to be great for our purpose. It has nice bands in layers 2 and 5 of the cortex. It's homozygous viable, the mice are normal. And then Sun-Chin Ha, a postdoc, picked up the project, and everything I will show you from now on is her work. And what we tested the reporter with the Relr mutation to ensure that if we identified defects of cortical layering, this strategy would work, and it worked quite well. So we entered the RGS4 mutant into our typical 3-generation screen for recessive mutants, and we were very pleased to find this abnormal mutant in which one of the cortical layers appears to have been lost. And then we did our usual positional cloning study, localized it very rapidly to proximal 13, which is exactly where the Relr mutation is. And indeed, what this proves to be is a splice site mutation at the very distal end of the Relr gene, which results in a C-terminal deletion, a small C-terminal deletion, and hence we call this Reln CTR-del. So this was a little bit more proof of principle than I had hoped for, but we actually I got interested in it when we examined the cerebellum in these mutants. And you can see the Relr cerebellum in the second panel and the CTR-del cerebellum in the third panel, and it appears to be perfectly normal. When we looked at other laminated structures in the mouse, or in the CTR-del mouse, what we also found is evidence for a hypomorphic allele. You can see, is this going to work here? Oops. You can see here, this is the normal layering marked by two different markers. Here's Relr, in which this is clearly disturbed, and here's our CTR-del, which appears to have some maintenance. I'll point your attention here to these neurons in the marginal zone, which ought not to be there. Another laminated structure in the mouse cortex is the hippocampus. This is wild type. Relr is profoundly distorted, and the CTR-del is missing the infrapyramidal blade. So this is a hypomorphic allele. One possible explanation is that this is just sort of reduced global function of the Reln protein, but the homozygous CTR-del mutant has the exact same phenotype as the compound heterozygote between CTR-del and Relr. Relr is a null allele, and if this was simply a dose effect, you'd expect the latter to be more severe, and it's not. It's identical. So what's going on? Relr is known to have its, Relr is a secreted protein. It's known to have its effect mediated through two different receptors, ApoE4 and VLDLR, and these have non-overlapping effects. So you can assess that by looking at the knockouts of the ApoE4 and VLDLR mutant, and you can see various aspects of the phenotype in ApoE4. There is no marginal zone invasion, but there is a perturbation of the layering, which you can't appreciate here in VLDLR. There is marginal zone invasion, and importantly, when you put these two mutants together, they recapitulate fully the Relr phenotype. So it looks like these are really the two important players in mediating Relr signaling. Similarly, for the hippocampal phenotype, wild type, you can see ApoE4 is affected, not that bad. VLDLR looks normal, but the double homozygote is severely perturbed. And lastly, the cerebellar phenotype, wild type, the two individual mutants, and then the double homozygote. So this enabled us to make a hypothesis. There were aspects of the CTR-del phenotype that sort of looked like that of the VLDLR phenotype. For example, the marginal zone invasion that's occurring in both of the mutants. And so we came up with the hypothesis that what CTR-del retained was activity through ApoE4, but had lost some activity or its ability to bind or activate VLDLR. And then this is a testable hypothesis using a genetic epistasis approach, where we would predict that if this is the case, if we combine CTR-del with the knockout, we should get a phenotype similar to the full Relr phenotype. Whereas if we combine CTR-del with VLDLR, given that we're hypothesizing this activity has already been lost, we shouldn't see anything that's too different. So Sun-Shin set up those crosses, and what I will show you here is that we see results that are not perfectly consistent with the hypothesis, but reasonably so. So here the Reln-del CTR-del combined with the ApoE4 mutant is identical to Relr, whereas the CTR-del with the VDLR null doesn't look too much different. Similarly, for the marginal zone, the cortical phenotype, ApoE4 does not have marginal zone invasion. The double mutant does. And finally, the cerebellar phenotype, this didn't really fit in with the model. The double Reln CTR-del homozygote is worse than either of these mutant alleles by itself, but certainly there is a cerebellum that's present there, which is not the case in reeler, and then the combination of real and CTR-del with the null, VDLR null, is even further puzzling because this actually, in fact, looks protective against the null allele itself. So we really can't answer that or explain that aspect of it, but we had enough evidence here that was consistent with the hypothesis to ask the biochemical question regarding the binding of the reelin receptor, the reelin gene to reelin protein to these receptors. And fortunately, there is a very nice assay that enabled us to ask this question, which was the original reelin binding assays. And what you do is you express reeler. It's a secreted protein. You take the supernatant from cells that are expressing it, and then you take the receptors that are expressed in heterologous cells, and you combine the supernatant, which contains reeler here, with the receptors, wash everything off, and then do a Western and see what binds. And that was actually very nice for our purposes, since enzyme or receptor binding is not our forte. And also what this does is this integrates all of the biology because it's all being done in the context of a cell. It's not an in vitro experiment. So we expressed the CTR-del in a heterologous cell type. What we did see is a reduction in secretion. And so what we wanted to do then to make sure we were comparing apples to apples is concentrate the secreted protein so that we have comparable inputs. And when we did the reeler binding experiment, we get the control wild-type reeler is bound equally to ApoER2 and VLDLR. And when we use our CTR truncation allele, you can see the very different binding effects there. So that's the story, the conclusions. Genetics works. And that's the view from my deck, unretouched. And this is Sun-Shin Ha, who did almost all the work that I showed today. Sun-Shin's looking for a job, so if you have need of a very competent neurodevelopmental geneticist, please let us know.
Any questions? I have one. Any idea what's going on in the cerebellum?
No. There is possibly another receptor out there.
But the the A and the V double has a phenotype, so that's, very interesting. Any others? Well, thanks for a nice talk, Dave.